An AI model developed by Stanford University researchers searched nearly 20,000 human genes and uncovered a tiny peptide hiding inside a much larger human protein. When researchers made this 12 amino acid molecule as a drug for animal tests, animals ate up to 50% less during the next hour, and obese mice lost fat over a period of 2 weeks. It is called BRP, but the most exciting part may not be the Ozempic comparison. It may be a new way to discover powerful biological signals already encoded inside our own bodies. So, first of all, let's see how this AI guided search works. The researchers built a system called Peptide Predictor. It scanned roughly 20,000 human protein-coding genes for secreted proteins containing cleavage patterns recognized by pre-hormone convertase 1,3, an enzyme involved in producing hormones including GLP-1, the appetite hormone. The search produced 373 possible prehormones and roughly 2,700 predicted peptide fragments. Scientists selected 100 candidates and tested them in neuron-like cells for biological activity. The strongest response came from a 12 amino acid fragment of the BRINP2 protein named BRINP2-related peptide or BRP. Mass spectrometry also detected it in human cerebrospinal fluid and mouse brain, suggesting that biology naturally produces the molecule. So let's see what BRP did in the lab and also in animal studies. Researchers synthesized BRP and tested its effect on feeding. In lean mice and Yucatan mini pigs, a dose given before food became available reduced intake during the following hour by as much as 50%. In a 14-day experiment, obese mice receiving BRP lost about 3 grams, largely from fat, while on the other hand, the control animals gained roughly 3 grams. The treated group also showed better glucose and insulin tolerance. The team did not detect signals of food aversion, nausea-like responses, anxiety, reduced movement, or major changes in water intake or bowel output under the conditions tested. That combination, lower food intake, fat loss, and no obvious aversion signal, explains the comparisons with GLP-1 drugs. But biologically speaking, BRP may be doing something different. The relation between BRP and GLP-1 may be a completely new biological pathway. BRP activated appetite-related neurons in the hypothalamus and other brain regions, but the pattern differed from GLP-1 drugs like Ozempic. It also appeared to act independently of the GLP-1 receptor leptin and the melanocortin-4 receptor. Its direct receptor remains unknown. Finding it could reveal a previously unrecognized signaling pathway used by the brain to regulate feeding and metabolism. And BRP may be only one example. The same strategy could search the human peptides involved in pain, inflammation, sleep, or immunity. A larger innovation is a discovery engine that treats our own proteins as a library of candidate medicines. So, at the end of the day, calling BRP a natural Ozempic captures attention, but it misses the most original part of the story. AI did not merely imitate an exciting weight loss drug, it helped uncover a biological message concealed inside a human protein, one that appears to influence appetite through a completely different neural route inside our brain. ERP may become a useful drug lead in the future, but the search method could be even more important here. Our genome may encode far more signaling molecules than current textbooks describe. PRP is still preclinical and has not yet been tested as a weight loss treatment in humans. Researchers must now identify its receptor, improve its stability, and test whether the effect translates safely. Did you like this video? Here in this channel of Science and Other Demons, I'm trying to talk about science, philosophy, and other stuff without the hypes. If you like such a thing, you might want to consider subscribing.